and a nice Monday to you. Welcome to RettSyndrome.org's virtual conference week. This is Paige Nuez, Director of Family Empowerment at RettSyndrome.org. And on behalf of our Board of Directors and team, we're so pleased to bring you this week's series of free webinars to elevate your understanding and deepen your knowledge of Rett syndrome research and care practices. Your children are very important to us, and having your attention engagement today demonstrates your deep commitment as well, and we're so glad to have you. Before we begin, if this is your first GoToWebinar experience, I'd like to share some brief technology points on how GoToWebinar operates. The audio feed is open only for our presenters, so you don't have to worry about your background noise interfering with the presentation. If you look to the upper right corner of your screen, you'll see a control panel window, and we invite attendees to write in your questions or comments in that question or chat box throughout the presentation, and we'll answer them as time allows, likely at the very end of the presentation. If we don't get to your question, though, during today's webinar, we'll work with our presenters to get all questions answered through email. And if that control panel window is distracting to you, you can click on the small orange box with a white arrow to minimize that window, and then you can click on it again to expand it if you wish to submit a question. Okay, so the next point is that this session is being recorded, and you'll be receiving an email with a link to the recording. If you're having trouble hearing, please check and turn up the volume on your phone or device. And we're going to invite you to sit back, listen, and learn. And I'd like to invite Janice Ascano, Grants Manager at RettSyndrome.org, to introduce our speakers and presenters today. Thank you, Janice.
Thank you, Paige, for organizing this exciting virtual conference week. This morning it is my pleasure in introducing our first two speakers, Dr. John McCall and Dr. Daniela Brunner. Together they will co-present the complexities of translational research, what it is, why we do it, and how we do it. Our first speaker, Dr. John McCall, is the president of PharmAC and will cover the path to new drug treatments for Rett syndrome. He has extensive experience in drug discovery and medicinal chemistry. He received his PhD in chemistry from the University of Wisconsin, trained as a postdoc at Harvard, and has held many leadership positions in the pharmaceutical industry, including Pfizer, Pharmacia, and Upjohn. He now has his own consulting and research company and spends much time on rare diseases. Dr. McCall is a member of the NIH Steering NeuroNext Committee and chair of the NIH Blueprint Steering Committee. He also serves on a number of scientific advisory boards for the NIH, biotechs, and foundations, including RettSyndrome.org. Our second speaker is Dr. Daniela Brunner, who is a senior scientific advisor of behavioral research and development at Psychogenics. She is leading the RettSyndrome.org Scout program preclinical testing and will cover the value of standardized preclinical testing using valid animal models and tests. Dr. Brunner studied zoology at Buenos Aires University in Argentina and later received a PhD in experimental psychology at the University of Cambridge in England. Dr. Brunner completed postdoctoral training at Columbia University, where she worked on animal models harboring genetic mutations and extended her interest of developmental biology to psychiatric disorders and the behavioral analyses of autism and schizophrenia. Dr. Brunner has also held faculty positions at the City University of New York, Hunter College, and Columbia University. In 1999, Dr. Brunner joined Psychogenics, where she expanded their animal studies and was responsible for bringing the Rett mouse model to the company. And in 2011, Dr. Brunner presented Rett mouse studies at the NINDS workshop called Setting Standards for Preclinical Research for Rett Syndrome. So we would like to thank you both, Dr. McCall and Dr. Brunner, for taking the time today to present the translational research session of this week's conference. And with that, it is a great pleasure to turn the presentation over to Dr. McCall.
Thank you. Let's see, show my screen. Can you see my screen at this point on the other side? Can you hear me? Can you see my screen?
We can, John.
Okay, good. All right. Danny and I are going to talk today about drug discovery and drug development using translational research. This is an exciting and complex business that I hope you'll find interesting. I got interested in Rett some time ago when I met Kylie Elser, who's the granddaughter of good friends of ours, Dee and Eldon Miller. And Kylie was a Rett's child, and really, since then, I've become very interested in the disease and reasonably active. In way of introduction, here's some information that you already know, I think. Rett's is due to mutation in the MECP2 gene, and I think of it as a neurodevelopmental rather than as a neurodegenerative disease, because you don't have neuronal death early in Rett's as you do with neurodegenerative diseases. And in a way, that's one of the things that encourages me about the disease. A lot of the hardware is still present in the girls. There's serious downstream consequences. Again, you all know this. When you have a mutation of MECP2, you have what are called dopaminergic deficits. You lose motor skills this way. They're noradrenergic deficits. This is involved with respiration and cognition. You get problems with what are called neuroexcitatory paths that have to be addressed. And then there are just effects, physical effects actually, on length of axons and dendrite density, which affects neural communication. So there's a lot going on. Yes?
John, sorry to interrupt you, but I'm not sure if you intend to be showing a slide right now. We're seeing a screen that says the GoToWebinar generic screen.
Okay, let's see. Well, that's probably... let me try to move my presentation over to the side. I have multiple monitors, which may complicate life.
There we go.
Does that help?
It is. We've got the slideshow view.
Okay, very good.
There we go. Thank you. Sorry for interrupting you.
Well, I'm not showing it as a slideshow yet.
No problem. In the meantime, I'll just remind everyone that in the questions field of the GoToWebinar control panel, you can type in any questions that you have during John or Danny's presentations today.
Well, let's. Can you actually see my slides at this point?
I'm seeing it with the timer.
Okay, this is problematic. It's showing as a trainer. What I need to do is change
better in the other. Shows better in the other screen. It's fine if you go in the other.
How do I change it to the other side, though?
Just what you did just a second ago, where we see the deck view. There you go.
There we go.
This will be fine. I can see it. If anyone is having trouble seeing, please let me know through the question or chat.
All right.
Sure.
Let's go. And I hope you can see the screen at this point. So, be right with you here. Okay. I'm talking again to the second slide, and what point I was going to make on this slide is there's a lot going on in Rett's disease, and that suggests many potential biological targets. And biological targets are, in a way, the feedstock for drug discovery and drug development. And what we're talking about today is translational research. And in translational research, what we try to do is move from the discoveries of basic research through a discovery process up to the time that we actually get the drugs into the girls and hopefully prove that they work. There are basically two flavors of translational research. The most complex is what I call from scratch. And from scratch, we begin with a biological target that we think is likely associated with the disease. And what we do is we try to find compounds that will, in a way, affect that biological target. And predictably, what we're trying to do is reduce or repair injury, correct a biological deficit, for example, the lack of a particular protein, or correct a biological excess. And I'll talk a lot about the actual steps that go into this from scratch in a moment. The second approach to translational research is repurposing. Now, repurposing is quite different because with repurposing, what we do is we take a compound that's been developed for another disease, but which hits a mechanism that we think is important in Rett, and we repurpose it for Rett's disease. Now, no matter how you do it, translational research is tricky, it's complex, it's expensive. It's definitely not a game for sissies, but it's the way we work. And indeed, no matter who does the work or where it's done, translational research follows a fairly set pathway. It begins, as I've suggested already, with the biological target that we think is associated with the disease. We then develop assays where we can find chemical matter. We pick the chemical matter that's most promising and we optimize that. I'll talk about this in a minute until we get to a compound we call a candidate. That single compound we take into man and then try to prove that it works in the disease. Now, the trend in major pharma is actually away from basic research and early discovery, and you've probably read about that, but the National Institutes of Health and foundations are picking up the slack in many diseases, and that includes Rett's, and that's fortunate. So there's still a lot of really good work going on, and I'll talk about that in a short time. Now, a candidate is the compound that's ready to go to the clinic, and it has certain characteristics that I'll just kind of go through right now. Pharmacology has a number of points, but what we're looking for is a compound that's potent, safe, and efficacious. And we measure things like what dose do we get an effect on in animal models of Rett disease, what's the tolerated dose, what's the no adverse effect dose, and so on. And this defines how the compound works in cells and in animals. On the chemistry side of things, again, this won't surprise you, we're looking for something that we can make at an affordable price. It has to be pure, preferably it's crystalline, it's soluble, and so on. And finally, we want to have a patent position on it so that we can interest the pharmaceutical industry in actually finally making and distributing the drug to parents and children after we register with the FDA. ADME is kind of a funny word, but it means absorption, distribution, metabolism, and excretion. And you really need to understand this because basically this is what happens with the drug after it leaves the stomach, goes into circulation, and where it goes after that. So a lot of work in the drug discovery business goes into understanding ADME. We have to understand what's called pharmacokinetics and metabolism. And finally, safety. The kind of drugs we're talking about are lifelong drugs, and so we don't want untoward effects that are cardiac or central nervous system diseases. We don't want a drug that's a mutagen that can cause problems in the oncology area. We're looking for compounds that can be taken really safely. Now, the drug discovery process goes through a number of steps, and it begins with hit identification. And I'm going to go through each of these steps. But basically, in hit identification, we develop an assay that allows us to examine the biological target that interests us. And then using that assay, we'll typically look at hundreds of thousands, literally hundreds of thousands of compounds to find compounds that seem to have an effect on the biological target. And this is called assay development and high-throughput screening. Once we have identified these hits, we test them in a variety of assays to see which ones are really believable and which ones seem appropriate. And the most encouraging ones we go into lead identification with. Now, here's another interesting word, SAR. That stands for structure-activity relationships. One of the important things in picking a type of compound we're going to try to optimize is demonstrating that when you change the structure, you change the activity either better or worse. So it means that it has the potential to be optimized. It's not a one-trick pony. It's a series of compounds. That's what we're really looking for in lead identification. So we do a lot of tests, and eventually we'll pick either one or two scaffolds, which is an arrangement of carbon atoms that we think are exciting. We then go to lead optimization. At lead optimization, there's a big, big jump in the amount of resources that are required because now we're optimizing on potency, selectivity, Does it hit just our target? Does it hit other targets? Is it safe? Can it be absorbed when given orally? Does it have a long enough life in the body to do what we want it to? Does it actually get to brain, which is important in most of the compounds in Rett's disease? So in lead optimization, which usually lasts 18 to 24 months when it's fully resourced, we eventually go forward and then we pick a development candidate. Now here's another concept that I wanted to introduce. We create what are called testing funnels. And in a testing funnel, we start after we make compounds, chemists make compounds, we want very high throughput, low cost assays that help us triage large numbers of compounds into smaller number of compounds. And that's called the primary screen. Then we go through secondary and tertiary screens, look for a lot of things as we optimize on chemistry. And this is a highly iterative process. As we learn things, we make new compounds that take advantage of those learnings. And eventually we move into in vivo studies. In our case, one of the models is the MECP2 mouse, which we think has a reasonable correlation to human disease, pick compounds that are exciting, and then we move forward to the clinic. Now, before you go to the clinic, you begin interacting with the FDA, and it's important at this point to have really pure compound. And so we develop what are called GMP, or good manufacturing process, synthetic procedures. We do safety studies. I usually do 28 days in a rodent and a non-rodent species to look for safety before we do any treatment of people. We look for cardiovascular, respiratory, and central nervous systems safety. We look for gene toxicity to make sure there won't be an oncology risk. And we develop various analytical methods so we can actually measure the compound when it's in people. And we eventually, at this stage, after we've done all this work, go to volunteers and we do safety and pharmacokinetics in volunteers before we go to children. Okay. Now, I mentioned earlier that there's a fair amount of retreat in large pharma, which is unfortunate but real. But that academic groups, small companies, mid-sized pharmaceutical companies, and foundations are responding. And this is really pretty exciting because there's a lot of smart and dedicated people now working in the field. And they are, as I say here, engaged in translational research. They're partnering with pharmaceutical groups, these are the foundations, and they're exploring venture philanthropy. In venture philanthropy, it's possible, for example, for a pharmaceutical or for a foundation to actually take an equity position in a small company under very generous terms and have a downstream interest in the product as it evolves. I have a company that is developing a compound for muscular dystrophy, and we've had very good participation with venture philanthropy with along our way. So this works very well. Foundations are also in the educational mode, of course, helping to understand the disease and sell the value of working in the disease, and also working with how to register drugs for therapies. Okay, what's happening in Rett's? This is actually pretty exciting. On my second slide, I emphasize that there are quite a large number of things that go on when you knock out MECP2. And very few are particularly good, as the audience knows, certainly even more than I do. But these are just 4 examples of translational research compounds that are in the works. Fingolimod is an interesting drug. It's called a sphingosine phosphate receptor agonist, long words. But the important thing here is it increases something called BDNF, which is brain-derived neurotrophic factor. And this, we know from a variety of work, is a protein that stimulates synaptic growth and strengthening. And there are clinical trials already going on with fingolimod. So this is an example of repurposing for Rett's. So there is a clinical trial underway in Europe now. IGF-1, this is insulin-like growth factor, and it's something I think that we've read a lot about in the U.S. This is a recombinant protein that promotes, it's called a trophic, it promotes cell survival, synaptic maturation, and it is in clinical trial. David Katz is working with groups of compound called NMDA antagonists, and ketamine's an example of this. And what he's seen in his mouse models is that these excitatory amino antagonists have dramatic and beneficial effects on respiration in the mouse. And there's a clinical trial that'll soon be underway with ketamine. And the last example I picked was serotonin, which is a strange compound. It's a serotonin 1A agonist and a dopamine-like agonist. And it addresses some of the dopaminergic deficiencies that we see in Rett's. And for example, John has shown that in 3 different models of Rett's, you get improved respiration with this compound. So those are examples of compounds that are underway now. So at this point, I'm going to pause, give the screen to Danny, who I hope will have better luck than I did in getting the screen shown, and we'll progress. If you have questions, we'll look forward to answering those. Thank you.
Hello, everybody, and thank you for joining us. I will try to explain a little bit how a mouse model can be used, and this is what John referred to. Once a compound is chosen, then and if there is a good mouse model, it's really imperative to use the compound in the mouse and look at in vivo function to make sure that one is prioritizing correctly what goes into the clinic, and also to make sure that the compound is safe in the particular disease that is being studied. In the case of Rett, I think there are several models that are very good. They do recapitulate many of the dysfunctions seen in the girls. And so I would like to show you a little bit how these mouse models can be used. The first thing I wanted to say is that the drug screen we set up is based on other drug screens that have been quite successful. What I call a phenotypic screen depends on having a mouse model of the disease, and in the case we use it for SMA, for Huntington disorder, and for autism. There are some interesting models based on genetic findings that one can use treating the mouse model, see if one can rescue their deficits. And at least in the compounds and the screens that were done at Psychogenics, at least 2 compounds are going or are in the clinic. One can also do library screening using normal wild-type mice and look for interesting signatures, and that's another type of screen that has been done in this company, and there are 2 compounds going to the clinic. There are different ways to pick up compounds, and some that John has mentioned. One can repurpose a compound that has been shown to be safe in a different disorder, or screen compound libraries, that compounds that are already synthesized and maybe something is known, but they have not been tested in humans, and then do the novel chemistry with, as described by John. For the SCOUT program that Steve Kaminsky put together for IRSF, with John's help and my help as well, the idea was to have, from all the compounds available, some that were mentioned by John, have a selection of compounds and prioritize them. From the compounds that have not been tested in mice, or in particular in the model of Rett that we are using, do an MTD. MTD means a maximally tolerated dose. This is a way to judge if a drug the dose of a drug may be chosen based on studies on a rat or a mouse or some other animal, but we want to make sure that in the model we are going to be using, the doses we choose for a long, very costly study are appropriate. So if we give a dose and we see a bad reaction, then we have a chance to go down. This is what this MTD phase allows us to start a more expensive study with a good dose. The HIT screen, or what we call the tier 1, is a high throughput, novel high throughput test or standard test in vivo, where we can see if we get any signal, any compound that could actually rescue any of the deficits in the mouse model. And then at tier 2, we want to replicate and expand what we have found in tier 1. So basically 2 tiers to make sure that the compound that comes out is the best compound available. So if the compound gets to clinical trials, then we know we did as much as possible to prioritize from all the things that we could be doing otherwise. So for a preclinical screen in vivo, we need to have a good rodent model, as I said before. And what is a good model is that it needs to have a robust phenotype. So the differences between a control mouse and the mutant mouse should be large enough that they can be rescued with a drug. So it's not just that it has a deficit. The deficit needs to be large and robust so we can actually try to rescue. We want to make sure that what we are measuring in the rodent has a strong neurobiological common mechanism with what is in the humans. And this is because this is a requirement to increase the translatability of the results in rodents and humans, which are very different, of course. So we want to do the best in the mouse to help prioritize the drugs. This is basically the point of a phenotyping study. Before we start, we did a lot of phenotyping for these models. One thing that we had to take care of is getting enough experimental animals. This may seem as a silly point. You just breed the mice, you have the mice, and now you can test the drugs. But this is one of the most difficult pieces of setting up a drug screen. First, that you want the mouse model to be a good one, that it needs to be available, and that you need to be able to generate enough experimental animals to test all the drugs that we want to test. It's a big challenge. We have solved this for Rett, and so in about 2 years, we managed to pick good tests that show very robust deficits in this mouse model. We have enough mice being bred right now at Jack's lab. This is a place, a non-profit, that specializes in breeding of mice of different kinds. So now we are up and running, and we are testing lots of drugs and getting very interesting results. So what I want to show you now is how these tier 1 tests look like. The first thing that we did is we chose the bird mouse. Many of you may have heard about Bert. Of course, he's a doctor who's done great things for Rett and created this mouse model that's really is a great tool. And it's available for anybody who wants to do research with. A lot of the early literature was done on male animals. The male mice are very sick, very small, and they don't survive for long. So the first thing we wanted to do is do a comprehensive phenotyping on females, because we believe that female mice will be better for translation into the clinic, because you have the 2 copies, or that you have at least 1 functional copy of the MECP2 gene. Indeed, the females are healthy, they live a long life, and as you can see in this graph, this is the body weight. They don't have any problems, so it's a very good model. And I'm going to show you now the deficits that we have measured and that we want to now rescue with drugs. The Rotarod is a test of motor function. Some of you may have heard about it, but this is, you can see a rod. This rotates. The mice go on top of the rod, and as the rod rotates, they need to balance and coordinate. And if not, they drop to the bottom and they stop the test. So we can measure how fast they drop as the rod rotates faster and faster. This graph shows you the latency to fall. In blue, you can see this is the bird female, and in yellow, the control mouse, the wild-type mice. And you can see the wild-type mice take much longer to fall than the bird mice. And we measure this at 8 weeks of age and at 12 weeks of age. We don't see in this model a very strong progression, which it fits with some of the clinical observations in the girls. We have other models where we do want to see progression, for example, mouse models of Alzheimer's, where things should get worse with age. So this is a model that has a relatively early phenotype, and it is more or less as we expected. As you can see, these two curves are very far from each other. The error bars do not overlap. So this suggests this is a very good endpoint measure that we can try now to rescue those in the bird female mice with the drug of interest. Another measure, this is very common in mice, is very easy to measure. If you leave the mice by the tail, the normal response is to extend the limbs, as you see on the left photograph. On the right photograph, they are clasping the hind limbs, and this is an abnormal response. And you can see here at 12 weeks, about 80% of the bird females do show the clasping, whereas about 20% of the wild-type mice show clasping. This is a very easy response, so it's part of the tier 1. Startle PPI, it's another test. You can see the chamber here is padded with foam, because what we do is we play a very loud sound of 80 decibels or more, and this startles the mice. They are on a platform that moves, so we can measure how much they startle. In this slide, you can see the startle is measured on arbitrary units here from 0 to 700. Wild-type mice startle about 500, and the female bird mice around 200. So this is a big, robust phenotype. We find it every time we test the mice. We see this as well. Other models of Rett have also been shown to have the same deficit, and this is published in the literature. So this is very robust. This is a nice endpoint measure that focuses on the circuitry. This is a reflex. So this is measuring neuronal function in a very simple neuronal circuit. And because we show already the mice have similar weight, we know this is not confounded with lower body weight, which is the problem when one uses male mice, which are very, very small. So when they startle, of course, one expects them to produce a lower response on this moving platform. So this is a very nice, very clean test, and in a very good, robust model. This is NeuroCube. It's a measure of gait. You can see the apparatus on the left top. It's we shine a blue light from the top, and also when they touch the bottom, and I hope you can all see the movie. So when they touch the bottom, you can see their paws turn red. So a computer can look at this video and pick up the red points and measure how the mice are moving. Are they moving in a symmetric way? Are they shaky? And that mouse that at the top of the top video, that's a wild-type mouse. And this mouse at the bottom is a bird. In this case, it's a male mouse. We have done this with females as well. And if you can see the video, you can see that movement is very shaky, very variable. So the mice do recapitulate the gait problems that are seen in the girls. So this is also a very good input measure. This is just an example of things that can be measured in using the system, not only where they put the paws and how fast they move, but also how the body moves from side to side. Is that normal? Is the position of the paw normal with respect to the center of the body? So we measure lots of things, and this is a very high throughput system that I set up in the company some years ago. The output of this gait analysis is done with computers. It's using machine learning techniques, which I'm not going to explain, but just believe me, they are very powerful. You can see here what we call the cloud analysis. The blue cloud represents the mice, the wild-type mice that we run as controls, and the red cloud are the bird female mice at 6 weeks and at 12 weeks. We can see a very robust gait deficit. If there was no deficit, these ellipses will be on top of each other. Because the two groups are very well separated from each other, then we know that the gait deficit is very robust. We can also investigate what kind of deficit we see. And the deficit is basically gait features and of how long the steps are, how wide the gait is, and also how the body moves from side to side, and importantly, how the paw is positioned, the angle of the paw. So we can get a lot of information by doing this gait analysis in mice. Gait analysis is done in other species, in horses and dogs and humans, of course. So it's very translatable and is a very good input measure. In Tier 2, we have 2 tests. We want to include a replication of anything that looked good in Tier 1, but we also want to expand. And we chose 2 tests for Tier 2. One is the optokinetic response. This is something that is done in the girls in the clinic. And that measures a reflex to follow a moving object. In this case, you can see the mouse in the middle of the apparatus. The apparatus is a cylinder that has these vertical bars. As the cylinder moves to the right or to the left at different speeds, the mouse has very subtle movements of the head, tracking the movement of these stripes. And then in this graph, you can see the number of responses. A normal mouse in yellow, a wild-type mouse, will show 8 to 10 responses when the cylinder is moving at low speed. At high speed, the responses are lower. So at low speed, we can see that the bird female mice have fewer optokinetic responses. So we believe this is also a very nice endpoint measure, which is quite different from anything else we are measuring. It's a very simple, robust reflex response. And finally, as you probably all know, respiratory deficits very important, and these mice do recapitulate the deficits seen in the girls. And this is something that we have tried to use a video system, but we finally decided to use plethysmography to measure respiratory deficit. The reason this is in tier 2 is because it's very time consuming and very expensive, but it's very important. So we do want to use it to look at any drug that passed the tier 1 tests. Finally, an overview of the SCOUT project. In the first year, we completed the phenotyping screen, and we chose the test that I just described. And now we are in the second year, testing lots of drugs, as many as we can. And this is what we are doing right now. We have a number of compounds which are in the queue. A number of compounds have been finished, and by the end of June, we will have a couple more completely done and analyzed. And at least one of these compounds showed a very interesting signal. There was a press release by IRSF and a press release by the company called Revive, which I'll show you the link so you can read what they say about this, the trial. I didn't want to go into too much detail about that, but it looked very promising. So we're very happy with the drug screen, and we're happy with these early results. I think this is my last, my last slide. And I'll be happy to answer any question you have about any of this.
Great. Thank you so very much, Danny, and thank you, John. I know that for many in our audience, this information is hard to extrapolate and correlate from the lab to our children, but what's very important for us that families understand your dedication to helping us. You know, it's a kind of a catchphrase in the industry right now to fail fast, and we want families to understand the real, very real steps of what that means when you hear that, fail fast. And the reason behind that are, really twofold. From what I understand from both of your talks, fail fast to me means, being very careful with dollars, because it's a very important indicator for both our donors as well as the companies that are developing these products, but also for time. And for us as families, that's the thing that we're most concerned with, is time. And getting these compounds through the SCOUT program and through these tests, to me, mean that it's a faster, safer way before we get them into testing our children. Can you talk a little bit about that and what the term fail fast means to both of you and your work?
Well, I'll start. I mentioned that the work that we do is complex and expensive. And what you would like to do is to eliminate compounds that aren't going to work in the girls as quickly as possible. You do that for financial reasons, but also because there are very few shots on goal allowed by both money and the patient population. So you want to be able to eliminate compounds that are poor, get them off the table, and make room for more promising compounds. So that's with cost, but also it's very strategic. Danny?
Yes, I agree. I think that even if I work with mice, the girls are in my head all the time, and I think it's very important that the clinical trials are done with the best drug possible. And it's not just a question of money; it's also a question of not giving false hope and not wasting time that could be used doing something better. So I think one has a moral obligation to do as much as possible to get the best compound tested in the clinic. And again, it's not just money. It's another. There are other reasons for that.
I think there's a misconception that the FDA scrutinizes at a therapeutic level what the chances of success are for a compound, but their real concern is with safety and exposure. And it's up to us to pick out the compounds at the candidate level that have the highest chance of success in going forward. Again, it's a relatively small patient population. It's an orphan indication. You don't want to have false shots on goal.
I think that's also important this webinar is very important because sometimes in the communities, the girls or the relatives, there's a lot of anxiety about getting things in the clinic as fast as possible, and so there is a conflict in going too fast and being too careful. So that one tries to have an equilibrium between these two forces.
Thank you. That explanation is very reassuring. Fail fast is a term that can mean, nobody likes failure, but in this case, it is good that we know what's going to fail before it reaches our children. And fast
could be rephrased as succeed quickly. It would be a little more appealing, actually.
I like that, John. Let's make that the coin of the realm. Okay, I have a question from our audience. If you'd both be so kind, I think this came up, John, when you were showing the slide about some of the compounds that are currently being tested. And you may not be the ones to answer this. This might be more appropriate for our panel on Friday about clinical trials. The question is, can you give an estimate of the time from when a compound passes the preclinical stage to when it reaches the clinic and could potentially be ready and in clinical trials?
Sure. Actually, I've just done this with a drug for Duchenne muscular dystrophy. So once you have the candidate, you have to work out a GMP synthesis and do the safety studies. And that takes 9 to 12 months to really be ready to go in. You then enter the clinic with normal volunteers. These are adult males, usually, not always males, but often. And we do what are called single ascending dose and multiple ascending dose studies. And for there, we look for safety and for pharmacokinetics, how long does the drug last in the body, and perhaps markers of efficacy. And from there, that's called phase 1. From there, and that's going to take a good year. And then we go to phase 2, and in phase 2, the initial phase 2 studies will be in the girls. And in the girls, you measure things like pharmacokinetics to be sure that they behave as the adult males did, and safety. And then eventually you can do proof of concept trials to show that the drug works. Now, if you're repurposing, and I mentioned repurposing where you're working with somebody else's compound that's been carried forward already, a lot of that work could be done. It's conceivable there could be an existing formulation that if it's already been in the clinic, there will be safety studies already done, and so you can short circuit the process and save a lot of money. Now, for the girls, a formulation that was developed for adults, for example, a capsule, might not be ideal, and you might want to go with a flavored suspension or something like this that might be better tolerated. But again, I use the phrase, it's not for sissies, and it really isn't, because it's fairly capital and time intensive once you actually enter the clinic.
Yeah, very, very much is. And we're very excited to have a few clinical trials underway. And another question that came from the audience is, and you referenced this, is the age for enrollment criteria for the drug trials. And I think this, if you wouldn't mind repeating some of the criteria that goes into assessing gender and age for these compounds once they get into human testing. Again, this might be better for the Friday panel. Go ahead and say that if you'd prefer that we hold that till Friday.
Well, it's clear that the original safety work, if it's a new drug, is done normally in adults who are volunteers, and we establish basic safety criteria, and usually in the multiple ascending dose, where you give higher and higher doses until you see a toxic effect. So you don't really see the girls until you've completed that work. And as I mentioned, if you're lucky and you're working with repurposing, somebody else has already done that for you. Sometimes you have to do pediatric trials, but the FDA has actually been very understanding with a disease like Rett's, the orphan diseases, where there's so few kids and the situation is so desperate. and they don't always require, for example, pediatric safety studies before you go into the clinic.
Thank you.
But once you go in, you don't want to have you want to treat a disease at a time when you could see a beneficial effect. So then that depends on what you're measuring. If you're measuring, for example, seizure frequency, then you're going to be working with older girls. If you're measuring progression of the disease or respiration, you might go with the younger group. So depending on what the indication is, you might use different age. But you don't go immediately into the girls with a new drug.
Thank you, John. Okay, I'd like to remind everyone, if you have any questions for our panelists, please type them in now and we can get them addressed. Having lots of comments from everybody about what hope they're receiving from this information and how reassuring it is that you're doing this work ahead of time.
You know, I mentioned neurodevelopmental. I work in a lot of different diseases, and I work in neurodegenerative diseases. The one thing about Rett's that I find pretty encouraging is a lot of the hardware is still there. You're not suffering early neuronal death like we do in some of the other neurodegenerative diseases. And really, that's, for me, that's pretty hopeful. I mean, it means you got a shot.
Yeah, quite a few more questions here. One of them is regarding the spectrum in Rett syndrome. And we know that, especially as in the younger girls and our women with Rett syndrome, the spectrum of symptoms is different across people and changes over time. Can you talk about how the drugs that you're looking at now might address those differences and how that works neurologically and biologically?
Well, when we set up the screen, we try to, as I said during my talk, we try to look at independent domains. We don't want to just focus, for example, on motor function and then find something that works on motor function but doesn't work in respiration. So that's the idea, that the screen is as wide as possible so we can capture any type of beneficial effect. So I think that maximizes the chances that a drug will get to the clinic and will have some beneficial effects for maybe a subpopulation, but that's something that John can answer better than myself.
Mm-hmm. I agree. And I think that I know on the Friday panel presentation, the clinicians will be discussing a lot about how the different drugs will be targeting different symptoms or not, in the case of perhaps correcting the very underlying problems that are happening with Rett syndrome, so that as the compounds work, that the symptoms themselves are not the target of the drug, but the outcomes will improve and they'll be variable based on child. And they're effective.
Yeah, they're basically two classes of drugs. You've got, the IGF-1 approach, the fingolimod approach, and actually we just mentioned a few of the other people trying to get BDNF directly into brain, where you look at a trophic effect that may improve synaptic function. And then you have other drugs which are really trying to treat some of the outgrowths of the disease. So anti-seizure meds, for example, the almost Parkinsonian-like symptoms that come with the disease, so the dopaminergics that people are developing. So you can treat aspects of the disease, or you can go after the disease itself. And the good news is both things are happening.
Mm-hmm. Great. I've got one more question that I'd like to bring up today, and then we can address the rest of the questions through a QA doc after our presentation. And if either of you could comment on the state of clinical trials and research around the world. Most of your work that you presented today is happening here in the U.S., and we have a lot of attendees that are in countries around the world, specifically in the UK and India, for this particular question. But I'm wondering if you can comment on how the process happens outside of the U.S. and if you're familiar with what's going on with Rett syndrome.
Well, I know that the dopaminergic work's coming out of Australia, but I tend to be U.S. and Eurocentric, and so I work in Europe and in the U.S. in terms of the clinical development of compounds. And one of the things that we try to encourage, and which actually happens, is cooperation between the EMA, which is the European Medical Authority, and the FDA. The process for moving compounds back and forth across the ocean is pretty well defined. So as far as doing the clinical trials, most of us know what's involved with that. But as far as what's going on in Rett's, I'm more familiar with what the drugs are, but not where the clinical work's going. So that could be a really good question for Friday. And once you get out of Europe, I mean, I know because the investigators, I know some of the investigators, but I don't know what clinically is going on in the countries outside of U.S. and Europe.
Mm-hmm. Great. We'll ask this question again on Friday.
That's a good question. I mean, that's an important question, really, because part of the problem with Rett's is it's a relatively small number of kids that we're trying to treat. And so you want to access as much of the population as you can. That makes it global. It's not just shown in a couple of kids that the drug works. You've got to actually show to the satisfaction of the regulatory agencies that you've functionally improved the child in order to get the drug registered, and that, of course, that it's safe. And that takes numbers.
Okay. Well, thank you very, very much, John and Danny. I'm going to close on. I know both of you are working so hard, and I have met you in person, and I know your commitment to our girls, and that means a lot to me, that your heart is really in this with your intellect and your brain and your resources, and we're so grateful to have you on our team. A parent did ask, what can parents do to help? And this is a perfect transition into my closing slide. RettSyndrome.org is working very, very hard to accelerate research, and we're doing that because of families doing fundraisers and donations, and with the partnerships that we're making in Washington to accelerate funding. We're able to do the webinars like these today at no charge because of our generous sponsors, Tobii Dynavox and Ameripride, and that enables us to redirect every dollar that comes into our foundation to the important work for our families, which is family empowerment and research. And research has the biggest price tag, and so that's where we try to put all of your support. And we thank every family who has held an event or is thinking about holding an event or has attended an event or has made a private donation. We are so grateful for your support. We hope that you found the value in today's sessions and that the knowledge you learned today will empower you in ways great and small in your path forward with Rett syndrome. We believe that information about our disorder should be made free and available to you so that you understand the timeline that we're working with and can trust that we're all working very aggressively but safely to help our daughters and our children. Whether it's correcting the underlying biology caused by the genetic mutation or deletion, or searching for that next medication or combination of medications that will prove in clinical trials to help symptoms, or if it's research that improves our ability to reset the neurology through pharmaceuticals, through therapeutic interventions, through an enriched environment, through improved AAC and adaptive devices, RettSyndrome.org is invested in all of these quadrants so that we can help our children today and tomorrow. And we know that our children are worth every effort to overcome the challenges before them. We know that, we see it in their eyes. And we're here to assist you with every challenge that you as parents and caregivers and guardians have before you as well. So we want to thank our speakers. We want to thank you for your support of our foundation. We hope to see you again in a couple of hours where we will hear from Dr. Huda Zogby on some of the basic science. That's the science that happens before we get these compounds into testing and in the translational arena. And I hope you take a look at rettsyndrome.org for the family conference to see all of the sessions that are being offered this week. We're going to go from research to practical symptom management and what our clinicians are looking at in terms of helping our girls today. And we'll also be doing some talks later this week on communication, devices, ways to communicate better with our girls, and how to help the partners on your team, therapists, teachers, educators, also learn some of these techniques. So please spread the news about these webinars. Invite everybody to register for them, even if it doesn't work with their schedules. By registering, you will get a link to the recording afterwards. And if you would, please take the time to answer the email survey after it comes out, and that'll help us fine-tune future offerings of this kind. We hope to continue in this format. We think it's really valuable, and we can reach people all over the world. So we appreciate our attendees for being with us today. Thank you for your fantastic questions. Thank you to our presenters. I hope we'll see many of you at our family getaway in Orlando this October, and we look forward to meeting you again in person at our rettsyndrome.org family conference next summer, June 24 through 26 in Illinois. So I hope that you'll plan to attend in person and get to meet folks like Danny and John in person. I know that they really like to know you too. Okay, thank you very much, and this concludes our webinar today.
Thank you.
Thank you. Bye-bye.
Thank you.